It's a pleasure to be here, seeing old friends and new friends from the UK and Israel. Today I would like to convince you that also very short, very small elements, very short peptides as well as metabolites could be of very important properties in physiology, pathology, and also technology. So, our approach for the last 20 years already was try to understand the mechanism of self-assembly, taking complex biological molecules, cut them to smaller and smaller pieces in a reductionist, non-biased way, and find very the most fundamental building blocks that could allow the formation of ordered structures. For many years, our claim to fame, as some of you know, it was dipeptides. In recent years, we've been interested in even smaller structures, including amino acid and nucleic bases. And why was there a lot of interest in those structures formed from such simple building blocks is the fact that you see very interesting physical properties, mechanical properties, optical properties, electric properties, semiconductivity, piezoelectricity, pyroelectricity, things that we usually think about elements from the silicon world, solid-state physics. I would describe it as solid-state biology. And another interesting point is that elements that we discovered, and I will speak about it today, in a very methodical and non-biased reductionist way, were discovered by nature and evolution well before. One example that I really like is that of guanine. We are all familiar with guanine as one of the 4 letters or 4 elements that compose the DNA and RNA. But guanine, at the cellular level, can form a structure like G quadruplexes, physical colors in fish, the ability of chameleons to change their color, reflectors in the eye of reptiles, and all by molecular self-assembly of such simple metabolite. We've been interested recently in the study of Tapita lucida. This is the reflective layer in the eyes of various nocturnal animals. In the case of cats, this is done by riboflavin. In the case of dogs, it is done by cysteine that is coordinated by zinc. It's quite interesting as, you know, cats and dogs, they show some hostility, but they are very close from the phylogenetic point of view. Lemurs that are much closer to us are using riboflavin once again. Using crystallography, I know that crystallography is now replaced by electron microscopy for larger structure, but still there are a very important role for crystallography, especially for small molecules. We could see how riboflavin could act as a reflector. Riboflavin has a very high refractive index in an anisotropic way, and you form a kind of a molecular mirror that allows the formation of the structures. And interestingly, until and now the structure was not known. Moreover, when we crystallize the riboflavin, we can understand the way in which the layer, the ordered layer, is being corrected by the physiological machinery, because this layer is always exposed to electromagnetic radiation. And so we simulate it by photobleaching, and then we see that there's lateral exchange of the riboflavin, and this is the way that it's been corrected. But I will get to metabolite at the end of my talk. I move back to, there's a review, because I don't have a lot of time to speak about it, so it's quite recent, and you are very welcome to read it. But how did we get into all of this? And this is lovely to have Sheena and Meital here, because our interest in very small structures began with amyloids. You had perfect introduction to this field. For us, these are self-assembled structures. And this slide is reflecting quite a lot of work, but in our reductionist, non-biased work, we went from quite large proteins. We were the first to show that pentapeptide and tetrapeptide can form amyloids, and later the dipeptides, diphenylalanine, can form ordered assemblies. In a paper published in Science already 17 years ago, we discovered that the entire information to form structures could be achieved if we cut beta amyloid, the polypeptide that forms amyloid in the case of Alzheimer's disease, to a region which contains a small element that could allow the formation of structures, and another two small elements that inhibits its formation. We show that just the unprotected amino acid could form nanotubular assemblies, and if we acetylate and amidate the termini of the peptide, we can form typical amyloid fibers. So this was the first demonstration that amyloid fibers could be formed by dipeptides. As many of you know, these very short peptide structures became very popular, mainly because, as I mentioned earlier, because of the physical properties. We realized already in 2005 that these structures are quite rigid. We measure it directly by using atomic force microscopy. So you apply a force and you can measure the point stiffness and calculate the Young's modulus of the structures. It was calculated to be about 20 gigapascal, which is like the mechanical rigidity of bone. Carol was mentioning some criticism by reviewers. We got a lot of criticism on this. Some people thought that we didn't measure it correctly. Later on, people working on amyloids, a few years later, discovered that indeed, in the case of amyloids, you can find this type of mechanical rigidity that is quite similar to bone. Other properties that we discovered are related to piezoelectric properties of the PNTs, which were similar to lithium niobate. I will get to it also later when I will speak about even shorter segments. And the advantage and disadvantage of working on such simple building blocks that immediately you have other groups all around the world, and especially in China, working on these peptides, and we're even making a device for energy harvesting that is based on the diphenylalanine peptides that we discovered. We also discovered the luminescence from these structures. Again, we met with some skeptical observation about it later on. It was replicated all around the world. And interestingly, it was discovered by the Cambridge group with late Chris Dobson, a mentor and role figure for me, and the Kaminsky group, that as we could see luminescence in the case of the diphenylalanine structures around 450 nanometers, that could be observed also with amyloid fibers. And other properties that we discovered about these very short peptide elements are semiconductivity, and again, I don't have a lot of time to discuss it, but I refer you to this review that we published about 3 years ago, 2.5 years ago, about the semiconductivity of the peptides. This is another slide from later time that reflects a lot of work trying to understand, going around the diphenylalanine motif, finding more than a dozen different building blocks that can form ordered assemblies, and of di and tripeptides, And in all cases, what we could see were things that reminds us of amyloids. They had the beta sheet-like structure. By that time, we know that they have similar mechanical rigidity as amyloid, similar luminescence as amyloid, and all like typical amyloids. We truly appreciated the significance of the phenylalanine motif, also based on a theoretical work done by Ulin and Tuttle, published in 2015 in Nature Chemistry, in which were ranked the aggregation propensity, the AP's aggregation propensity of different tripeptides, all the 8,000 combination of dipeptides, 20 by 20 by 20. And you could see the list here. It's diphenyl, the FF motif is very common, PFF, VFF, FFM, FFF, and the others. Those who are not with diphenylalanine motifs were with tryptophan, and some people did not read carefully our science paper from 12 years before that, but we could notice there also the role of the tryptophan. So all what we discussed in 2003 was found here. And then but still we could not understand what is why, let's say, proline PFF has higher propensity to form structures as compared to, let's say, FFF, which is more hydrophobic. And using crystallography, we like crystallography, we wanted to understand this. We started by seeing whether structures could be formed, things that looked like amyloids, and we could find things that looked like typical amyloids, and we knew by that time that we could form amyloids by dipeptide and tripeptide. But it became quite interesting when we were using biophysics. So looking at the CD spectra, it looked helical. Looking at the Fourier transform infrared, it looked helical. And then crystallography showed us that indeed, very strangely, for a very short peptide motif, we get helical structures. And at that time, it was already the paper, and thank you, Meital, for the introduction, the beautiful paper of Meital in Science. And we could see something that could remind us alpha, how you call it, alpha helix, cross alpha amyloid, but with such simple peptides. So I do agree with you that it might be more common than we appreciate. And this is, so peptides are still full of surprises. Playing around with peptides, but at that time we are now, could well establish the fact that you can form structures that are very rigid mechanically. In the case of the helical structure, you get extra strength by having this hydrogen bonding by the helix organization. So we are getting into 44 gigapascal, which is already metallic-like structures. And we always like to be inspired from nature, and thinking about helical structures that give mechanical rigidity, you always think about collagen. And in the case of collagen, instead of a proline, you often have hydroxyproline with an extra hydrogen bond. And indeed, we could get from Young's modulus of about 44 gigapascal to Young's modulus of over 100 gigapascal. So from self-assembly peptides, no covalent bond beyond the basic building block. We could get things with mechanical rigidity similar to titanium, just by the molecular self-assembly. So it's so until 7 years ago, I could keep speaking about peptides, and they are wonderful. Many people, our group and many other groups are now more than 200 groups around the world working on diphenylalanine and its derivative. and making all kinds of very interesting technological utilization of it. But we wanted to see what is the real definition of the smallest element that could form structures. And we got into a completely new world just by doing a simple control. So I told you that we work on diphenylalanine, we work on all kinds of phenylalanine derivatives, we worked on a triphenylalanine, and again, until 7 years ago, if somebody would ask me what is the minimal requirement to form amyloid, I would say that the dipeptide, because of the MI bond, you have the resonance in the bonds, it's partial, two-dimensional in its nature. But we're experimentalists. We wanted to see whether a single amino acid, phenylalanine, could form all the structures. The answer should have been known, and this would end the story. However, when we tested phenylalanine assembly, we could see things at relatively high concentration in the millimolar range that look like amyloid, that bind THT-like amyloids, that bind Congo red and show this typical green apple, golden apple birefringence between coarse polarizers. And we scratch our head. On the one hand, this was formed in relatively high concentration of phenylalanine, not physiological whatsoever. On the other hand, it looks very much like amyloids. And also, we show the typical electron diffraction like amyloids, and so on and so forth. But we then realized that there are some pathological conditions, not physiological, in which you have very high concentration of phenylalanine. So the phenylalanine concentration for, I guess, everybody in this room is around 40 to 60 micromolar, even if you have a very high protein meal. But people with phenylketonuria, it's a disease in which individuals lack the enzyme that converts phenylalanine into tyrosine. If they are not on a very strict diet, the phenylalanine concentrations could reach the levels of millimolar. Actually, the clinical definition of phenylketonuria is phenylalanine concentration over 1.2 millimolar. So it's more than 20-fold higher as compared to normal individuals. And if you look about the symptoms of phenylalanine PKU patients, if they are not well treated, you see things that reminds us of neurodegenerative disorders. And we ask ourselves whether it's possible that indeed phenylalanine assembly into the structures is related to the neurodegenerative phenomena that we see there. And we could see indeed that phenylalanine could not only form ordered assemblies, but it could form toxic assemblies at the concentrations above 1 millimolar, similar to the pathological conditions. We could raise antibodies toward these structures and pull down and inhibit the toxicity of these structures. And then we sent it for publication. We had tough referees, which was very good, because they told us, well, it's only circumstantial evidence. Please show it in animals. Luckily, for us as scientists, in the case of mice, they have exactly the same amino acid metabolism pathway as humans. And so mice, PKU model mice, could show the spontaneous evolution of antibodies that could recognize the structures that we identified. We send it back to the journal, and then we say, oh, okay, it's quite convincing, but now show it in human patients. Luckily, I had a very talented PhD student, now a faculty member at our Lee Adler-Abramovich, now a faculty member at our Faculty of Medicine. And she found in the Netherlands a brain bank of deceased post-mortem patient of PKU, and we could show for the first time the formation of deposits, like we see in Alzheimer's disease, of the phenylalanine that was co-stained with Congo red and with antibodies. So we now could show the reductionist scheme also to here. Again, the advantage and disadvantage of working on very simple system is that immediately you are being tested by the entire world, and you get a lot of interesting ideas from different groups in the world. Carol introduced us the beauty of mass spectrometry, and immediately after we published the paper, which we suggested that phenylalanine organization should be like that, was work by Mike Bowers from Santa Barbara, who, using ion mobility mass spectrometry, actually told us that this is the case in high pH, but in neutral pH, which is the relevant one, the organization is like that, which is fascinating, because if you look at the organization of the structure under neutral pH, because of the sweeter ionic property and the charge state of the phenylalanine, you have electrostatic interaction between the building blocks, which forms the core of the motif of the assemblies, but all these very hydrophobic side chains are exposed to the solution, and then you can understand how such structure could become toxic and interact with membranes. So we are convinced about the beauty and power of ion mobility mass spectrometry, and we try to apply it now to other metabolites that we study. So indeed, phenylalanine is one example in which you have very high concentration of a metabolite due to a genetic disorder, but there are many other unfortunately, there are many other what is known as inborn error of metabolism disorder, in which you have the accumulation of various metabolites. So, as we like, in a systematic, non-biased way, we studied all the different, list of all different metabolites. And indeed, we could see that not only phenylalanine, but also other small metabolites, adenine, orotic acid, cysteine, tyrosine, uracil, all can form things that for you working on amyloids look by electron microscopy just like amyloid and bind THT and Congo red. Moreover, now we become even more biologists, and we studied not only the toxicity, but the mode of toxicity, and we could see that the toxicity is being achieved by late apoptosis, which is what you see with proteins and peptide amyloids. So it's also behaves biologically like proteins and peptide amyloids. And interesting, and until now it was mainly in vitro. A very interesting example to show it in vivo came from a collaboration with ophthalmologist in Rambam, Shiri Zaytsuda is here in the audience, in which, starting from clinical observation with a patient with hyperoxaluria, with a high concentration of oxalate in their body, could see retinal abnormalities, but the puzzling observation was seeing retinal damage without the formation of calcium oxalate crystals. And here it's summary of what we did, and this is the retinal damage as observed clinically in the patients. We could see that also in the case of oxalate, we could form oxalate fibrils and inject it back into rat and see the same clinical observation as could see with the patients. So this was just published recently. It was an indication that you can get it also in vivo model. Knowing that we can form amyloid-like structures by different metabolites, we've been interested, and understanding the similarity to protein and peptides structures, we've been interested to see whether amyloid inhibitory agents that inhibit protein and peptide amyloids could be effective also in the case of metabolite structures. And indeed, we could see that polyphenols like EGCG and tannic acid, which are well-known modulator of amyloid formation by proteins and peptides, could affect also the formation of amyloids by metabolites like adenine and phenylalanine. An other interesting point came from what I mentioned about the physical properties of the peptides and protein and amyloid structures. So I told you about the luminescence that we saw with the diphenylalanine assemblies. Later on, the Kaminskis in Cambridge, in collaboration with Chris Dobson, could see with proteins and peptides, and we could see it published recently. We could see it also with the metabolite. As could see with those structures, we could observe a clear luminescence with adenine, tryptophan, tyrosine, phenylalanine. You could really see that it's fluorescence because of the fluorescence lifetime. So it's not just a reflectance, but rather real fluorescence with all the properties. And this relates to what we discovered with the diphenylalanine and the amylase. So I think that we get more and more pieces in the puzzle to show that structures that are made by the metabolites share properties together with the peptides and protein assemblies. This is also true for the mechanics. So I told you about 2005, 20 gigapascal for the dipeptide structures. Later on, similar numbers for the amyloids. And now, fighting with referees, hopefully we could show that also in the, we can show, but hopefully we could publish soon that in the case of the phenylalanine fibers, this is in collaboration with Nicholas Reynolds in Australia, using Q and M AFM, we could see once again also in this case the Young's modulus of about 10 gigapascal. Another thing that I told you about was the piezoelectricity, and here, not by us, unfortunately or unfortunately, we like that other people work on it as well, published only 2 years ago. You think about all the time that people were working on amino acids. Only 2 years ago in Nature Materials, describing the piezoelectricity of amino acids, in this case, glycine in its beta form. So I think it's what we see, it's a continuum, as I said earlier, a continuum in which we see the biological properties, the physical properties of amyloids made of large proteins and peptides, by ultra-short peptides like dipeptide and tripeptides, and by metabolites sharing all the same properties. And how can it be? So metabolites appear to be quite different from proteins and peptides. So we think, and again, crystallography comes to our advantage, and again, quite surprisingly, the first crystal structure of sweeter ionic phenylalanine only in 2014. If you think about amyloids, they have the beta sheet structure. We go with the zigzag type of arrangement. What we see with the phenylalanine packing, we see something like a supramolecular beta sheet, in which you have, again, the zigzag-like arrangement, but not with covalent binding between the monomer, but with non-covalent binding and hydrogen bonding between the different building blocks. And we see that it's quite general phenomena for various amino acids to form such layered structures. What are we doing in recently? Trying to understand the metabolite aggregation and amyloid formation. We're moving more and more into biology, at least the biology that is not too complicated for me to understand. We're moving now to yeast models. Trying to understand, for me, it's like the early days of protein aggregation, trying to understand what is happening there, what occurs, what kind of mechanism does the cell involve when you have the accumulation of metabolites. The first model that we used was of adenine accumulation, and we could see the sensitivity of a yeast cell to the feeding, external feeding with adenine. Usually when you think about the growing of microorganisms, they grow better as you give them more nutrients. But in this case, we replicating what is happening in the case of the patients which accumulate adenine, we blocked adenine salvage, and then in a dose-dependent, nonlinear way, until some kind of concentration, a given concentration, they can handle it, but from some threshold, very steeply they lost their ability to grow. Correlating the inability of the yeast to grow with the formation of aggregates. Another thing that we could see is our ability to rescue the growth of the yeast by the use, again, of polyphenol, in this case tannic acid, but also many others. And very interestingly, what we could see is the fact that we could be in a situation in which we provide the polyphenol, in this case tannic acid. We are still in a situation in which there's very high concentration of adenine in the cell, but due to breakage, disintegration of the aggregates that are being formed, we can rescue the cells, which give us some hope about translating it to human patients in the case when you have high concentration of metabolites and you can somehow disintegrate structures and allow a normal phenotype in spite of the high concentration of the metabolites in those individuals. We've been also interested to see the interplay between metabolites and proteins, because we also still have their affection to protein-based structures. And we suggested in some kind of hypothesis paper that protein, that metabolites may seed the formation of amyloids by larger proteins. And we had some reasons to believe that this is the case. In some neurodegenerative disorders, you see a situation in which you have elevated concentrations of metabolites. For example, in the case of Parkinson's disease, you have elevated concentration of quinolinic acid, and we could see that quinolinic acid by itself form amyloid-like structures, and this is electron microscopy and the crystal structure. And when you incubate these structures that are being formed together with alpha-synuclein, some of the people in the audience are working on Parkinson's disease and alpha-synuclein, which is, it's amyloid-forming proteins, but not in a very efficient way. But if you had the seeds of the quinolinic acid, you get much faster formation of amyloid structures. So this may serve as some kind of seeding mechanism. In the last 5 minutes that I have, I want to share with you our technological thinking about learning from nature and being inspired from nature, and ending up with one of the examples that of the guanine that I mentioned in the beginning. So we've been working on peptide technology and nanotechnology, as I said, for the last 2 decades. And we were very much convinced that peptide nanotechnology is very important and could allow a lot of technological applications. But in parallel, there's quite a lot of work on DNA nanotechnology. And we decided that since we like peptides and we also appreciate the specific organization of nucleic acid, to try to combine the two by the use of peptide nucleic acids. We did not invent them, as many of you know, they were invented in Denmark in the 90s as some amino acid analogs that allow the control or interference with genetic information expression. And these are, actually it's not peptides, but peptoids. They have a polyamide backbone, and they have side chains, unlike phenylalanine, alanine, glycine as we know with the amino acid. We have the side chains of the of nucleic acid. We like to synthesize. We don't like, we like not only crystallography but also synthesis. So we and we like doing things in a robust, unbiased way. So we synthesized all the 16 different combination of dipeptides, because we knew that dipeptides could form all the structures. And boom, we saw that only guanine-containing structures, building blocks, could form all the structures, which take us to, again, to this slide that I showed before. And with crystallography, we could see that what we envisioned really happened. What we see with peptide structure, we see the stacking of structures, one on top of the other, to form a pile, like you see with amyloid structures. But in the case of nucleic acid, you have the specific hydrogen bonding between the bases. And what we see here is the combination. We have the basic building blocks is folding upon itself to form stacked structures, and then you see the hydrogen bonding, like Watson Crick hydrogen bonding, resulted in a very dense structure. When you get new structures, you get new physics, and we could see something that was quite remarkable. Again, starting with as a control, we wanted to publish it highly, so we wanted to show that it has DNA-like properties, meaning it could intercalate some dyes, and we did the right control, and we check it also without the dye, and we could see fluorescence also there. This wasn't new for us because we could see fluorescence with very short elements, but we could see something that is known as red edge excitation shift. The emission of the fluorescence was depending on the excitation wavelength. So this structure could be blue, green, yellow, orange, or red, depending on the excitation structures. This is a phenomenon known in inorganic material, in glassy materials, but we see it now very vividly in biological materials, probably because of this very dense structure, the combination of stacking interaction and hydrogen bonding, as we see in the, with these structures. Just to finish on this, you know how it goes in the lab. While people were working on the dipeptides, di-PNA, other people in the lab were working on the structure formed by metabolites. So we asked ourselves whether a single amino acid, PNA amino acid, could form structures. And we studied protected PNA building blocks. Only guanine-containing structures could form self-assembled structures. Very uniform one. See this, it's not silica particles. It's just these building blocks. And all you do, you take the powder, you boil it and cool down, and you get this perfect seamless spheres. And you can also make an array of the structures. We are always sensitive to physical appearance. So we could see immediately that these structures have nice color. Luckily, by the time that we wanted to publish it, there was this beautiful paper about the way in which chameleon changed the color. by changing the spacing of guanine crystals, and we made something like an artificial chameleon using these structures. One last thing, Meital convinced me to put it, and this is for the student. If you have an idea, put it in writing. Maybe 15 years later, somebody will do something with it. When we just discovered that such simple building blocks like peptides could form ordered assemblies, we say, wait, and we could see the interaction of the structures with the nucleic acid. We say maybe it could have been relate to the origin of life. Ada Yonath was mentioning origin of life, but at much later stage. What is the element? How can you start with things that you find on meteorites, forming in a Urey-Miller experiment, and form ordered assemblies? So you could find amino acids, dipeptides in those assemblies, and we thought that it might be related to the origin of life. Ronald Riek from ETA took it many years later and do wonderful work on the role of peptides in the origin of life. But my message is, if you have some idea, put it in writing. We did it quite often, and it was quite useful. So my time is ending. We were hearing today about beautiful and complex structures like the ribosome and the photosystem, which are extremely useful, but also in the simplicity, there's some sophistication. I hope that I convince you that very simple peptides, metabolites can form ordered assemblies. They have role in physiology, pathology, nanotechnology. I would like to thank all those agencies who are supporting our efforts over the years. This is our group in a winter day in Israel, and this is to convince you that we like chemistry. So thank you very much.
Thank you very much, Ehud. So, questions?
Joe.
These, first of all, it's very interesting, Ehud. Thank you very much. But did you ever think of, these are probably all optically pure compounds that you've been working with. Yeah. Did you ever try to mix?
Yes.
To see what would happen? And would that in any way be possibly related to origin of life? Both. But first, in terms of materials, I'm curious.
Yes. So it's an excellent question, and we do study both the pure structure as well as racemic mixture and different ratios of the structures. We speak a lot with Meir Lahav. It's wonderful to see people who studied amino acids in the 80s and 90s. That things are getting, you know, as I showed earlier, the piezoelectric of amino acid published in Nature Materials in 2018. There's a lot to discover there, but you are completely right. We could see in the case of the racemic mixture some enantio separation between the structures, and this might be related to the origin of life. As you may know, there was a lot of discussion at the Weizmann Institute about the different abilities of various enantiomers to form structures. We try to keep ourselves out of this controversy, but we may get into it if we don't have a chance.
Did you find any interesting properties from these racemates?
Yes, we find, especially when you have co-crystals, sometimes you have stronger structures, more hydrogen bonding, and structures that are mechanically more rigid. Another thing, I didn't plan to speak about it because we didn't publish it yet, but I will mention it as a teaser. We work now with Rona Aman at the Weizmann on the chiral-induced spin selectivity, and we see quite remarkable effect of different, and also with Yossi Paltiel. the brother of Michal. We see beautiful effect of various chiral structures on these properties.
Perhaps I could ask one. Yeah. I can ask one question. So that is, obviously, the nanotechnology applications are probably very more futuristic and very interesting, but the properties are very interesting. Are there macroscopic applications? Like, for example, I remember William Astbury knitted a sweater made out of polymers that had been, or perhaps fiber reinforced plastics. Are there any people interested in that angle on your work?
Yes. So we were. At least we could convince the European Research Council to give us a proof of concept extension of our ERC grant on making artificial skin in which you are using the piezoelectric properties. For me, living in this interface between biology and technology, I'm always thinking, what is the meaning of this for biology? Because what we discover now, and it was known for many years, but things like collagen are highly piezoelectric. And you have other structures that in biology, is it related to tissue regeneration? Is it related to some orientation? So many of my students are pure chemists, material scientists, and I try to push them back to my biology. And also, as was mentioned earlier today, looking for a disease in which you have some changing of the properties. So this isn't as you rightly asked. It's on the one hand we are using the properties to make real words, artificial skin, reinforced structures, and so on. But I'm always amazed by the way in which biology was making all these mirrors, lenses, piezoelectric structure, conductive structure without the use of metals, only in the ionic form, but not of solid metals. And we learn how to do it with organic building blocks.
So I don't see any more questions. So if not, let's thank Ehud.